Awesome. Okay, so without further ado, let me start. My name is Theodore, I go by Theo, and today I'll present to you spatial and single-cell metabolomics. We'll discuss technology, we'll discuss a few applications, and then we'll focus a little bit on challenges. I work mainly at EMBL, which is European Molecular Biology Laboratory in Heidelberg, Germany, but also I'm very fortunate to start creating another company. And again, as Daniel said, I had one in the past, I co-founded that one. So it was a very interesting experience. And now we're actually commercializing single-cell metabolomics technology at the Bioinnovation Institute, which is an incubator and foundation in Copenhagen, not far away from where you are. So before I'll present everything, I actually would like to present and acknowledge and share a few words about the team. So this is the team in Heidelberg, and here we combine biologists, mass spectrometrists, bioinformatics and computer scientists, and systems biologists, and also devoted software engineers who help us develop software that I'll be showing later, and which is used by many people throughout the globe. And so you see, it's very interdisciplinary. This is what really helps us develop these methods close to applications. And the team in Copenhagen, you see here, it's a bit more corporate. And so because it's more focused on commercialization, and there is also a number of talented and very diverse expertise and individuals here as well. If you're interested to get in touch with us to learn more about any of these aspects, just feel free to contact me afterwards. So in terms of our topic and our focus of our research over the past years, it got crystallized on metabolic cell state. And we know that it's very dynamic and also very elusive phenomenon, because cells, they can change their metabolic metabolic state within under a second, because of very, very rapid turnover of metabolites. But also it is very affected by various factors, not only DNA and cellular programs, which are used definitely to govern this, but also non-genetic factors like cells, environment, disease, of course, reprograms the metabolism. And also there is a lot of interest how to develop therapy that would capitalize on the knowledge of metabolic cell state. And this is one of the focus of our commercialization activities here. So what we do, we develop methods and technologies for spatial and/or single-cell metabolomics to really get rid out close to phenotype, to resolve it single-cell heterogeneity, and provide information as much as possible in situ and in the native context. So we do it by using mass spectrometry as the main tool for detection. So we developed a lot of data management and data processing and data analysis software. And on top of this, more and more we're getting into the topic of using machine learning and AI to really extract information out of this. So now if we'll start talking about why anyone needs spatial single-cell metabolomics, so then lots of people, they have lots of different answers to this. I'm showing those which are closest to our heart and to our ambition and vision and plans. So one is tumor microenvironment. And here it was very, very interesting to follow the advances in single-cell transcriptomics and spatial transcriptomics currently, where there is a lot of different cell types discovered there, but not only cell types, but also cell states. And many of these states, they actually associate with metabolism. And on top of this, there is a lot of non-genetic factors here, which are hard to capture with sequencing-based technologies, or even antibody-based technologies for protein detection. So, and these factors are, this is acidity, because we know that cancer cells, they secrete lactate, increase in acidity here. It's also hypoxia, it's also metabolic waste. So there are discussions, and there is a lot of interest to understand how these gradients, non-genetic gradients that metabolomics will be able to capture, how they actually affect the tumor microenvironment, and how they sometimes prevent the therapy to succeed here. And the second topic for our attention and interest is spatial roles of immune system. And here I'm showing review from our collaborator, Erica Pierce and colleagues, where they discuss these immune cells, like they compare them to nomads, which are born in one place And afterwards, they get attracted to this place. They squeeze through the blood vessel wall, and then they get inside. And here they need to reprogram their metabolism in the solid tumor. And for this, they actually adapt in their metabolism similar to cancer cells. So, and if we can understand this process, and if we can characterize this, and we can even help immune cells in these adaptations, then we can make them more fit, and we can create better cells that will be more effectively fighting tumor cells. So there is a lot of interest in basic science, but also in therapeutic applications in either of these approaches and topics. So how do we develop methods? What kind of technology we use? MALDI imaging mass spectrometry is our underlying technology for pretty much all methods that we are currently developing. And this is mass spectrometer equipped with some sort of spatial sampling. Here it is being laser. And that's why MALDI stands for Matrix Assisted Laser Desorption Ionization. Here you see our setup here in Heidelberg. Our setup in Copenhagen looks different. So there are different sorts of those imaging mass spectrometers. But in this one, the laser is sitting on top. Then there is through the set of mirrors, it gets propagated here, where we take a tissue section for cells. We put them on a glass slide like this one. Then we put them into this chamber. We close the lid. And what happens next is a laser will be firing at a particular location, and molecules will actually be desorbed. There will be a sort of plume of molecules created here above the tissue section. This plume will get captured into mass spectrometer and then analyzed there. And for every pixel, we'll get a spectrum that will be representing the relative abundance of the molecules at this location. This spectrum will be collected on MS1 level. Tandem MS or MS-MS is actually not very common and very hard to achieve, in particular because of the limitations of the material that we get from one pixel. So then what we can do, we can select one M over Z for MS1 spectrum. We can take the intensities for this M over Z across all pixels, map them back into their locations, and get an ion image. And this ion image will be representing the relative abundances of the molecule of interest with this M over Z, or other molecules with the same M over Z across the whole tissue section. And now we can perform metabolite identification, and we'll focus on this a little bit and get the molecules which are behind these images. So long story short, the technology, the cutting edge, commercially available technology can perform analysis with 5 to 10 micrometer pixel size, can detect a lot of molecules, about say 100 small molecules, about 500 lipids, can detect drug metabolites, generates a lot of data because we can get tens of thousands of pixels, that means spectra, and this is high resolution mass spectrometry per section. And over the past decade, this became the major technology for spatial metabolomics. This is sort of images that MALDI imaging mass spectrometry can generate. Examples here with a 10 micrometer pixel size, showing molecules of different molecular and metabolic types. For example, here, adenine, some modification of amino acid, citrate, and small lipid. And we can detect more in one shot, and we can overlay them, and you see that they represent at least some anatomical structures of the tissue. But the key problem in the field of the past decade, similar to bulk metabolomics, was metabolic identification. And this was till recently our main project. And here we developed an engine called Metaspace, and we put it also on the cloud on this website, Metaspace2023.eu, where anyone can submit their data, can get their molecules back, can get their images back. This happens all within about 10 minutes. And they can also start sharing all these data sets in the browser, and they can also do additional analysis there. So long story short, this became a big platform and supported by European Commission and also NIH, because there is a lot of funding here to make it work for cloud computing, but also for developers and also R&D. This is how it looks. If you go to Metaspace2023.eu, then you can find a lot of public data sets, a lot of projects showing these public data sets, showcasing or through publications. So here I'll pick one, which shows whole body sections from animal models. And here we see on the right, a typical imaging mass spectrometry data set, where here it was collected of the whole red section. Here on the left, you see ions, which were annotated in this data set. And for every ion, we show what is the adduct, what is additional information like MZ, what is the score, what is FDR level, which corresponds to the confidence of annotation, and additional information like structure and many other things. And since it's imaging, there is also visualization capacities here that one can use. So moving on, over the past, I would say almost decade by now, Metaspace became a pretty large community-driven platform. So we have a lot of submissions, almost 40,000 submissions. And what's very cool that 9,000 of them are public. It means there is like 9,000 public data sets there for anyone to look at or use. It is used by many users. There is a lot of publications coming out of this. And it's also very collaborative for us, but also other people use it for collaborations because you can share data sets. You can use it through API, through Python API, and so on and so forth. So it's growing very, very nicely, and we are very, very happy about it. So wrapping up, so summary of the first part of my talk, that metabolism emerged as a key factor in biology and medicine, and you know probably even more about this, and you have your own perspectives on this. Spatial metabolomics, if you're interested, we can dive deeper a little bit into the history, but what's important that it actually maturated, and imaging mass spectrometry now is used mainly for spatial metabolomics, not for, say, proteomics or glycomics. And now it's really in the stage of development where it's spreading now beyond the early adopters' labs. In Metaspace, we have developed this big data infrastructure for spatial metabolomics and understanding metabolism. So this is the summary. Let's now talk a little bit about computational challenges. And 3 years ago, I have summarized this in this review, that this is the pipeline of data analysis, data processing, starting from signal processing to visualization, stat analysis, molecular ID, and molecular validation interpretation. And this was the focus of computational development 3 years ago. And it's pretty much the same, but there are a few changes here. So, I mean, these topics are still very important, but there is more and more interest and demand and gaps here in molecular identification, and also interpreting all this data, but not from statistical perspective, like clustering or like differential analysis, but really making molecular sense out of this. So, and for this talk, I put a few challenges which I think now are the most pressing, but also the most promising for anyone who would like to study and work on those. I think molecular identification is still very challenging. So, because first of all, we analyze data on MS1 level. Second, because of how mass spec is organized, we don't have this continuous flow of ions, so we cannot really do a lot of things like in standard LC-MS. But there is one very interesting approach here that is experimental coming up, which is ion mobility separation, which sort of can be run online and provides additional dimension for separating the molecules similar to LC. So an integration of ion mobility separation into molecular identification, maybe potentially with machine learning, can be a very, very promising approach in spatial metabolomics. There is still a lot of things to analyze for spatial and image analysis. And I think the most challenging and interesting and underexplored is molecular colocalization. How do we actually calculate it? What would be the measure even to calculate it, and all different approaches. Molecular interpretation, in particular, enrichment of pathways and classes. This is one of the few things that we are focusing on. In particular, because since we do identification on MS1 level, we have inherent level of ambiguity in our molecular assignments. So how this ambiguity propagates to enrichment analysis, this is a very interesting question. Big data analysis and data sets integration from multiple replicates, but also multiple studies. I think this is an underexplored topic. So there is quite some interest on batch correction, but actually there are approaches beyond that. Multi-omics becoming very, very important topic, also with experimental developments for co-registration, for pixels deconvolution, and for molecular integration. And standardization now is becoming more and more pressing because the field is really growing now very fast and spreads again beyond just a few labs for the early adopters. So this is about spatial metabolomics. Let's now switch gears and discuss single-cell metabolomics. And this will be the second part of my talk. And in the single-cell metabolomics, if we look historically, we check the first papers, then already 10 years ago, 2013 and 2011, there were two papers, one in Science and another in Nature Methods, which were discussing single-cell metabolomics like it exists, like it works, and how to do profiling, like perspectives and so on. But what's very, very interesting that if we look at the publications, and this just reflects what we know from our knowledge of the field, is that till pretty much very recently, maybe 2 years ago, or maybe even 1 year ago, there was almost nothing happening here. Single-cell metabolomics, almost no publications in this field. So why? Because those early pioneers, like Jonathan Swidler and also Renato Zenobi, they really did amazing work, tremendous studies and so on, but actually single-cell metabolomics was out of reach for others, for pretty much most of us, because of the lack of sensitivity of mass spectrometry. So in here over the past decade, the sensitivity of mass spec, in particular commercial mass spec, pretty much almost out of the box, got so high that now we can do single-cell metabolomics on commercial instrumentation. And definitely MALDI imaging mass spectrometry also contributed to it, because apparently it's actually maybe easier than using ESI, electrospray ionization, for this analysis. So it's now a rapidly emerging field. So 2 years ago, it was recognized by IUPAC among top 10 emerging technologies in chemistry. So last year, we had the first ASMS Asilomar conference, which united single-cell mass spectrometry approaches, including metabolomics as well, but also proteomics, lipidomics, glycomics. And now there will be actually a new society if you're interested on this. And there will be annual conference also on this. And early this year, single-cell metabolomics was recognized by Nature among 7 technologies to watch in this year. So we are contributing to this very, very hot topic and emerging field, in particular the method SpaceM. The method was developed by our first PhD student here out of EMBL, Luca Rapes, was published a couple of years ago. We also patented it, and this is what we're commercializing now in the incubator in Copenhagen. So the method goes like this. If we take the cells, we either grow them on the glass slide, or we can plate them on the glass slide. Then first we do microscopy of the cells to really find where they are, but also find their morphologic, fluorescent phenotype, and anything that we can get out of microscopy. And we take the same cells, we put them into imaging mass spectrometer, we run the data through Metaspace to do this molecular annotation. We get these pixelated images of metabolites or lipids or drugs or drug metabolites detected, but this is not single-cell data yet. Luca has figured out how to overlay this data with a subcellular precision, and also how to perform pixel to cell deconvolution and normalization. And now we can assign intensities for every metabolite or lipid or whatever we detect to individual cells. We can also represent this data as a matrix, where for every cell we have intensities of metabolites, we have intensities coming out of microscopies, such as fluorescent stainings, morphometric features, size of the cell, elongation, and spatial features, which cells are next to each other. And once we have this matrix, then we can actually start analyzing this data, and I'll showcase it for the study of non-alcoholic steatohepatitis, which is a common factor of liver cancer and this intermediate stage of fatty liver disease. And this was done together with our collaborators, Matthias Heichenwälder and Mira Stadler from German Cancer Research Center. And Mira, since then, she moved to our commercialization team in Copenhagen. And here, if we look at microscopy images of the cells, and we stain them with lipid staining shown here in yellow, then we actually can see that in this in vitro model of NASH, which is human hepatocytes stimulated with free fatty acids and also pro-inflammatory cytokine TNF-alpha, we see that some cells, they show this phenotype of accumulating lipid droplets, and others they do not. And since then, we see a lot of similar examples of single-cell heterogeneity in other contexts as well. So here we work with Matthias, Mira, and Luca, and everyone to really understand what are the molecules, what are the lipids which are accumulated in these lipid droplets. And for this, we first took hepatocytes, we did microscopy, we put them into MALDI imaging, into SpaceM, and then did signal processing, data analysis, and I'm showing you a UMAP plot where one dot is one cell. They're organized here based on similarity of their metabolic profiles of 700 metabolites and lipids. And we see here as a population of cells, which actually, if we overlay now, on top of this molecular information, if we overlay microscopy information, and here it's being fluorescence of this lipophilic staining, then we see that cells in this population, they actually have very high lipid droplet accumulation. So these are those red cells which have a lot of lipid droplets. So we can find now individual molecules which have high intensity of markers of these lipid droplets. And this is what we did. Here we calculated for every lipid, one dot here is one lipid, correlation with this lipophilic fluorescence. And those cells here on top, they will be the strongest markers or the most accumulated in lipid droplets. We can do enrichment analysis, we can find the function of these cells, and so on and so forth, or these lipids and so on and so forth. But we also did another step in this study where we considered slightly more complicated experimental design, where we took control hepatocytes which are actually model of fatty liver disease, but without inflammation. Then we added a pro-inflammatory cytokine IL-17A. Now this is in vitro model of NASH with inflammation. And here we added anti-inflammatory molecule TPCA1. And now if we do the same UMAP analysis, one dot here is one cell, they're color-coded here by their condition, we see that hepatocytes which are unstimulated, they're different from stimulated ones. And even more interesting, if we do clustering and we now compare this cluster to this one, then we see single-cell heterogeneity, where, for example, yellow hepatocytes, they also reside in this red cluster, and not all treated hepatocytes, they are moved away from this inflamed state. We can also find the pseudo-time trajectory and interpret all these shifts along this trajectory. For example, here, shift from this cluster to this one would be metabolic mode of action of this anti-inflammatory treatment. We found the trajectory computationally. We aligned all cells along this trajectory, all 30,000 cells. Now we can find the markers which are expressed at the end of this trajectory, and we can validate these markers by here comparing single cell data with a here bulk lipidomics from livers of mouse model of NASH, and we can appreciate how well the markers that we found in the single cell analysis, how well they actually reproduced in bulk livers. And many of these markers were found to be relevant in the human context as well. So wrapping up this part of the talk about single cell metabolomics. So I would like to share the take-home message that it is an emerging field. So we have developed a method for spatial single cell metabolomics, which uses MALDI imaging spectrometry, which uses pretty much commercial instrumentation, and all the secret sauce is in computational analysis and in the software. So this method reveals metabolic states in steatosis and NASH. And since then, we actually developed a lot of additional things for this method. We increased the throughput. We modified the experimental protocol to have small molecule detection, because previously it was demonstrated mainly for the lipids. Now we have it working not only for adherent cells, but also for T cells, which is very, very interesting in the context of immunology and immuno-oncology. And just recently, we also demonstrated 13C isotope tracing. We just put a preprint on bioRxiv if you're interested. So in principle, almost anything can be done with this method because it uses commercial instrumentation. It also uses MALDI imaging, which is a field being developed on its own. What kind of challenges here? So I'll go on very high level for single cell metabolomics. So signal processing is still a challenge compared to, say, spatial metabolomics, where it's much less of a challenge by now. So here, there is a lot of questions which we don't know how to answer, in particular in assigning pixels to cells. There might be different approaches because one pixel can sometimes sample several cells. It's also normalization, scaling. All these questions become very, very important in single cell analysis. Then data analysis. And here, we're learning a lot from single cell transcriptomics and other single cell omics, but there is a lot of specific questions here for differential analysis, for this and for that. In particular, because the data that we're collecting is very sparse, and the model of the noise might be actually different from single cell RNA-seq. And this brings this to very, very interesting directions of challenges. The data imputation, how to actually impute the missing values. And even if you don't need them for your specific analysis, actually working with sparse data might skew your statistical analysis. So you want to have somehow address the challenge. And data and noise modeling, so it's also a question which is very theoretical, but might help addressing this and this question. Multi-omics data integration becoming very interesting here as well, in particular in light of a lot of interest and parallel developments in single-cell RNA-seq. So how can we integrate it in terms of on the level of conditions? How can we integrate on molecular levels? So standard, and then there are also two big challenges: standardization. So the field is so new but it's already becoming a problem, for example, to share the data because everyone is using their own format. So, and the topic is being discussed and definitely will be addressed. And now we are lacking common data repository. In spatial, metabolomics, Metaspace is one of these examples. It's probably like the biggest data repository right now, but it also can be shared through metabolites and other repositories. But for single cell metabolomics, it's very, very challenging because the data is slightly different format. So the repositories absolutely not adapted to this in terms of visualization, metadata, and so on and so forth. So these are all challenges which can and should be addressed. And with this, I would like to wrap up. I would like to say thanks to the whole team who contributed at EMBL, then also collaborators, then past members, and everyone who helps us at EMBL to perform this work, and lots of funders. And here you can see also our last retreat, which was joint retreat with a Copenhagen team, really, really nice. And I'm looking forward to have any questions from you.
Awesome. Thank you so much, Theo. This was a fascinating tour through the spatial world of metabolomics. I think the floor is open for questions. So if you want to ask Theo something, feel free. I'm happy to relay in case he can't hear you well. Apologies for the sound issue. Yeah, we have a question over there. Sorry, can you say it again? Yeah. Okay, Theo, the question was, what are the smallest spot sizes you get with MALDI?
Yep. And Daniel, thanks for repeating the question because I couldn't hear it. Okay, the smallest for the commercial instrumentation, it is about 5 or 7 micrometer in diameter. So in terms of ablation, but also in terms of the size. So there is a lot of developments which are not yet commercially available, which have demonstrated ablation spot as small as 2 micrometer or 1 micrometer, and even below that one. So I think 600 nanometer was demonstrated the smallest one for MALDI. However, the challenge there becomes not even the pixel size per se, but already sensitivity, because sensitivity, amount of material definitely decreases quadratically with the pixel size. So, and already with 5 micrometer, we're approaching the sensitivity, at least with detection of small molecules, and limit of detection for many small molecules. So in parallel, there is a lot of experimental work being done to increase the sensitivity. And here there are interesting approaches like MALDI-2 and so on and so forth. So, but yeah, commercially, it is 5 to 7 micrometer diameter.
Right, thank you. Okay, Theo, one question that I was wondering, so during the summer school here, we talked a lot about like LC-MS data, which is, I think for many people, the tool of choice for metabolomics. How different is like the coverage of like this molecular space between like single cell metabolomics with like MALDI-MS versus a standard LC-MS approach?
Yeah, so it's a good question, and I don't have a definite answer because there is surprisingly few publications doing this analysis. I can tell that we, in our experience, and some of this work will be published soon. So we see something like, maybe I would say like 1/3 of overlap between MALDI and LC-MS if we go for pretty standard LC-MS for untargeted. But there is like in each of these technologies, there is parts of molecules which are uniquely detected. So roughly 1/3 in MALDI, roughly 1/3 in LC-MS. It definitely depends on your protocol, on your method, and so on and so forth. I'm talking about the same polarity, right? So there is a lot of kind of like complementarity here. And some examples which I can mention, which might be, for example, easier for MALDI, but surprisingly not easy for LC-MS, is citrate. I was showing you this molecule like in for the brain tissue section. And okay, we cannot really resolve citrate from isocitrate, so because they're complete isomers, but at least we have detection. And in LC-MS, it's actually a very tricky molecule to detect. So, yep, but this will be like a high-level answer, and for more comprehensive, we actually need to have more studies published, again, by us, but also by others.
All right, cool. Do you see any use of LC-MS for single-cell metabolomics anytime soon, or is this already a thing at all?
Oh yeah, no, it's LC is tricky. Like in general, what people do for single-cell metabolomics, and this is actually where some of the early approaches, where like you take a cell, you sample the cytoplasm of this cell with sort of micropipette, and then after you analyze this micropipette by some highly sensitive mass spec. And this highly sensitive mass spec can be like direct infusion. It can be something like nanomate, where you even enhance the ionization through some sort of nano chip. Or it can be nano LC-MS. But you really want to go like not even micro LC, but to not like close to nano LC, because the volumes is very low and you can't really use your standard LC for detection. People do this. So the problem there is you need to go to nanoflow, and it's very, very tricky to use in metabolomics. So it's not very robust, not very stable. So I think that with the improvements in nano ESI for metabolomics, there will be actually more people doing something like microsampling or laser capture microdissection, where you cut out a cell using laser and then you subject it to the analysis. So yes, I think it will be actually more of this work complementing MALDI in the future.
Yep. So do you think like single cell proteomics is paving the way for this?
It's interesting. I think single cell proteomics is definitely more developed, and it's very interesting that now they are actually going on streamlining. A lot of work right now in single cell proteomics goes into streamlining analysis and also in computational protein ID, and because there are also some challenges there. So in this respect, I think that for streamlined experimental approaches and the cell sampling and cell sample preparation from individual cells, yes, I think they will develop a number of methods, microfluidics or any sort of like low volume liquid handling that will be useful for metabolomics. Mass spec is, however, is pretty different. And we see that, for example, nano AC and nano ESI that was is like the most common thing in proteomics didn't really translate to nano AC for metabolomics. So there is also some unique steps that I think we need to do as well here.
Okay, thank you very much. There is also a question in the chat, I see. Do you have any personal experience with MALDI 2 and TIMS or ion mobility, and in general for the detection of lipids or like peptidic compounds? Are there any, I think, like is MALDI favoring certain like compound classes there?
Yeah, so MALDI 2 and TIMS, we are just starting with this in Copenhagen. So previously we didn't have access to this instrumentation, now we have. But we're just starting, so I think it's a bit too early to say. What's interesting is that we have just helped Shane Ellis from Australia to publish one paper where they show that MALDI 2 works not only for lipids, as it was published earlier, but also for small molecules. I think the paper was published pretty much this week, so you can check it out. But we just helped a little bit with computational analysis there. So it means it seems like MALDI 2 works, but one should be very careful about like for which molecules, and also do we already have unwanted fragmentation? There might be. Ion mobility teams, I highlighted that I think it's very, very promising, but we have very little experience right now. And I think here is also one should be very careful about what kind of value it brings, not to really oversell it. Detection of non-lipid compounds. Yes, we actually, over the past couple of years, we worked quite a lot on detection of small molecules, because in particular for these questions of immune cells and immune oncology, there is a lot of interest in metabolic states like glycolysis or respiration of immune cells. So we work there, and yes, we do by now, I think, quite some good experience. Secondary metabolites, we didn't work much on this, in particular because secondary metabolites, they're often, if we're talking about secondary metabolites in the host pathogen, then secondary metabolites of the pathogen, they're normally like, it can be in very low concentrations, so detection can be tricky. So I worked a little bit with Peter Dorstein, and then afterwards here as well, and I helped some other collaborators with the secondary metabolites analysis for agar plates. I think this is very interesting field. There is not much going on there, but I think there will be more. Yep, and drugs. Yes, we do have some analysis. Actually, MALDI is pretty good for drug detection. That's why it's used in a lot of pharma companies nowadays.
All right.
So can I
cannot actually, in the meantime, I can take a couple of questions very quick. APS MALDI stands for atmospheric pressure MALDI. And another question for detection of single lipids or something from single microbes. This is very, very tricky because they are very small and maybe because of properties of the membranes. So we tried it, but we didn't succeed. So I think it's, you really need to be like, do something special experimentally to do single cell lipidomics for microbial cells.
Okay, I think we got another question here from Robin.
Yeah. Hi, Theodore. So about MS2 fragmentation spectrum in MALDI, and like there's not so many papers being published with that, right? And maybe just because there's not so many methods just readily available, like data dependent acquisition or something. So do you think we just have to push the vendors of the machines to develop some new methods there, or how can we get more methods?
Okay, so it was a combination of a statement and a question. So Robin thinks that there's not too many MS2 methods out yet for MALDI imaging, and he asks you how we can push the vendors to develop more such tools, or what is your take on this?
Yeah. I'm not sure it's actually like the problem is really pushing the vendors here, because most of mass specs for MALDI imaging, they actually are configurable enough in order to run your mass spec method as you want it. So even including like fragmentation, the problem is much more foundational. And the problem is simply that, like, if you have one pixel, then you pretty much need to decide what you use the ions from these pixels for. So it's hard to impossible to use ions from one pixel for MS1 and MS2. So, and it's not like, you know, like in LC, you have constant dilution of your analyte, and you're just sampling it, and you're like, oh yeah, so this is my like peak for MS1. I'll take top 10. I have already some exclusion list or inclusion list, and I'll take those. Or you do the data independent analysis. So here you really like, you always need to like, you either sacrifice your spatial information and you will use it for MS/MS, then you will not get MS1. So, or you'll use it for MS1 only. So, and this is what people do. So you can do, for example, MS2, but if you want to collect it broadly information from any pixels, then you need to sacrifice spatial resolution. For example, it's pretty easy to configure mass spec so that it will be like in sort of checkerboard manner. It will be like one pixel MS1, next pixel MS2 for one precursor, this third pixel MS2 for another precursor, and another one MS2 for the third precursor. But then it's only 3 precursors, and then you're sacrificing your spatial resolution twice. So, and this is the challenge, right? This is the challenge. So you actually need to be very smart about choosing your precursors. And that's why normally people do it only for, say, drug imaging in DMPK studies in pharma, where, okay, you want to absolutely be sure that this is the structure of the molecule. And then I care maybe only about drug and a couple of metabolites. Okay, easy. And there is actually Waters, they have just released imaging triple quad system. And I think this will be very interesting where it will go, because their limitations are even more apparent. But then on the other hand, there are also advantages. And for specific applications, will be definitely a very, very useful instrument. So, so long story short, it's not really about vendors. It's really coming up with a smart way of what you want to use MS/MS for. And likely where the field will go, unless there will be something completely different, is that field will go to using MS/MS for validation. There will be sort of like, you run it on MS1 level, then you do some analysis, ID, then you probably do statistics to really find the markers that you are of interest to you, maybe like 5 or 10. And on those, you either on the same tissue section, maybe on another one, you actually run MS/MS in sort of data in targeted fashion. So this probably where the field is now going. But there is always possibility for some crazy jump in sensitivity, or for some clever design for mass spec, where you actually cycle your ions for MS1, and then afterwards you use the same ions, you transfer them into some sort of CID, you do fragmentation. This definitely will be awesome.
All right. Thanks so much for your answers and vision on the future of MS2 in MALDI imaging. I don't see any more questions here, so let's give Theo again a big applause.
Cool. Thanks for the invitation, and enjoy the rest of your summer school.